Many of you emphasize Roy John's contribution to practical science and translational science, but I have to emphasize that Roy was also a discovery scientist, and this is the part of Roy John that I'm mostly connected to. Besides being a scientist, he was also a facilitator of collaborations, especially reaching out to countries like Hungary when I was behind the Iron Curtain. He helped us, especially my mentor, in multiple ways, and I try to follow his attitude as well as his pioneering aspect of being a good diplomat. And he always emphasized that scientists are the best diplomats, and the world should be led by scientists rather than by politicians. Following that, I have organized a meeting in Havana, Cuba with Pedro and Mitch. I traveled Iran and Tatarstan, and in my laboratory currently we speak 14 languages. So this gentleman here, is an unassuming person who's looking into the void or into the future, inspired us in many different ways. But for me, the first real interaction with him was this paper that I read, that at the time I was a medical student, but I was already under the influence of Skinnerism and especially Pavlovianism, and how memories are formed. And in this review, he attacked this idea of connectionism and came up with an idea that is embarked on Lashley's distributed system idea, and that had a tremendous influence on me. Namely, he thought that there are endogenous processes, there are many things that are distributed, and he was searching for tools to understand this, how you can read out something which is endogenous that I call internally organized. Now, I have to make a statement here that I'm a little bit biased because my own mentor, Glenn de was also trained or collaborated with Roy John. He spent 2 years in his laboratory, and this is the paper that I think eventually pinned down what components of the evoked responses could be related to endogenous or self-organized activity, and which ones are related to the physical attributes of the signal. Now, basically, the idea is that the longer the latency, the larger the components, the more time the brain has to distribute activity. So by the time we're at the P300 in humans, then a very large part of the brain real estate is involved. Therefore, it can make conscious decisions or conscious evaluation of the environment. So these are waves. These are secondary or tertiary replications of what's going on in the brain. So I spent the rest of my life trying to tease out the electrophysiological unit-based or single neuron-based explanation for these things that introduced by Roy John. And this is a necessary tool in order to understand feats of humans like this one here. I give you a moment to enjoy. Something that you have to put in your memory. You absorb the visual world in this case, and then you replicate and use your internally organized activities in sequences in order to generate actions. So you can see the fast activity in one after the other, that there is no other way to think about how it was possible to make, except that there was a memory-guided action sequence. So I've been looking for sequences for many years in my life, and here's one good example. One way of addressing internal organization is to keep everything stable, to keep everything constant relative to the external world. So we designed this very simple experiment where the task of the rodent is to go to the right or go to the left. If the animal finds food on the left, it has to come back to this wheel, run for 15, 20 seconds, keeping in mind that the reward was on the right. Now I have to go to this other side. It's a spontaneous alternation task. While the animal was running and other parts of the experiments, we were recording from neuronal activity in the hippocampus. Now, each line here is a series of action potentials, and the color code corresponds to the activity of neurons. We, the experimenters, made a sequence of activity based on the animal's left choices. And you can see there is a nice trajectory. The lifetime of each neuron is about 1 second. And then whenever this lifetime is over, it gives that information to the next neuron, the next neuron, the next neuron, and there are several hundred neurons here. So in this very nice sequence, you can see that this sequence is very different when the animal makes a left choice or when the animal makes a right choice. You don't need to do the hell lot of statistics to appreciate that, and that you can see that even online, sometimes even with a single neuron, we can predict whether the animal will go to the right or left, including errors with 90% accuracy. 15, 20 seconds ahead of the animal's physical choice. So this is a very powerful demonstration that indeed internally organization is very, very useful. Now, this is online, so to speak, when the animal is running in the maze. In this case, it's a simpler one. The animal has to run from left to right, and there are rewards on both sides. Now, these cells are called place cells in the hippocampus, and this is why they are sequentially organized. And you might say, in the spirit of Roy Jones' introduction in that paper, that all of these neurons are controlled by the constellation of the environment. The reason why the red neuron fires here is because it's activated by a chair in the room, and this one is by the corner, and so on. But the funny thing is that of course it's very difficult to assume that every single spike in the hippocampus, many synapses away from all sensors, will be guided by the external world. And in order to zoom in even further, we found that prior to this running, the animal is sitting immobile. The brain state is different from theta. This is called the sharp wave ripple state. And the same neuronal sequences are played out in a forward manner. The animal finishes the journey, it runs to the end, and then again, it becomes immobile while it's drinking. And the same neurons are again replayed, but in reverse. So this very simple demonstration shows that, there may be a planning process, a very fast process lasting for about 100 milliseconds before the journey starts. At the end, the animal can recapitulate what it learned, and that's what happens. Now, it turns out that, of course, this brain switches not only during waking from active to resting state, but also during sleep. So from this and other observation, we came up with what we call the two-stage model of memory. In essence, the idea is that during waking, the information comes from the neocortex to the hippocampus, and during offline states, the information goes back from the hippocampus to the neocortex for memory consolidation. We have about 2,000 to 4,000 sharp wave ripples each night in humans. So there are plenty of subconscious possibilities to rehearse aspects of what we have learned during the day. So stage 1 is learning, stage 2 is memory consolidation. And again, the sharp wave ripples are the key to this. These are packages, very powerful synchronous packages in the hippocampus that are responsible for this consolidation. So this is what we have known. Many laboratories have done experiments that support this idea that indeed, in order to remember things, we have to consolidate them, and the consolidation process takes takes place in sleep. So that's nice. We can go from learning to memory consolidation. But at this meeting, you will hear many talks, and only a few snippets will be remembered. And these snippets go into your brain because the brain selects. So the question is, how from the myriads of possibilities that come into our brain, the brain will select a few. So we are after figuring out what is the mechanism of this memory selection. And I'm going to explain it in the next couple of slides. So the first thing we can do is, of course, as Roy John always did, the technical developments. So now we are capable of recording from hundreds of neurons simultaneously, which gives you a very high resolution picture of what's happening in the brain, and it can be read out. The ground truth can be read out by the animal's behavior. So this is a maze, and you can see the animal's position is tracked very nicely, and the activity of the neurons that are recorded simultaneously also track the animal's position in the maze. This is not new. People have done it in various ways, and the Nobel Prize was given for this. But this is a much higher resolution, and it gives us a possibility to go even deeper. So you can see that the pattern from trial to trial is very, very similar. So the idea has been around that when things happen in a similar way, they are really identical in the brain. But if you zoom in a little bit and look at individual neurons, this is why it is important. It's not looking at the en masse activity, as Roy John had no other choice in those days, just had to look at the integrated activity of millions of neurons with the LFP and the Hawk potential. But now you can see that the individual contributions of neurons are very skewed. Some neurons are very important, others are less important. So here is one example that this neuron on an early trial fired quite a bit, but on the late trials, it's almost silent. This neuron actually changed its place field, and this neuron was the other way around. It was lousy at the beginning or didn't fire at all, and it fired quite a bit at the end. So there are these tiny granularities. There are drops on the tsunamis in this mechanism in this network activity, and the task is to identify the drops riding on the tsunami. One way to do that is take snippets of time, let's say 100 millisecond time beams that correspond to the theta cycle, and project this n-dimensional space into a two-dimensional thing that you can see on the right. And you get some interesting, funny, but structured pictures. And we would like to go back to the ground truth, which we can, because our ground truth is the animal's position in the environment. So what you can see here is that we go from the maze trajectory to this neuronal manifold, and you can imagine, or you can see that the animal starts here. This is the middle arm, and then whether the animal will go to the right or left, very early on, it's separated in the animal's brain. And so then we go one step further, because, as I say, it's almost trivial from the previous research, but what we ask now is not where the animal is, but in what trial the animal is running. In other words, which lecture your memory is in now, or which lecture you are going to remember. So this is another UMAP showing that from 1 to 70 trials, you can see that the color very nicely separates trial 1 from trial 2, and so on and so on. We do one more trick here, a little bit that I don't have time to talk about, but basically what you can see is that trial separation is also possible from the activity while the animal is running. Now, that's interesting in itself, that the brain keeps track of order. But the interesting thing comes now is that what happens when the animal is rewarded at the end of the run, when the sharp phase really occur? What is being displayed? What the first trial, the second trial, the fifth trials, or any of those? And the answer is this confusion matrix very clearly shows that it is always the immediate or the surrounding experience that is being replayed during this waking sharp wave ripples. Okay, so sometimes there is no ripple at the end. Other times there are many ripples. So there is an opportunity that certain events are replayed more and more and more time, and those experiences are those that had happened immediately before or immediately after the change from theta activity to sharp wave ripple activity. So now we have a template here, an interesting template showing that not each of these experiences are guarded or classified equally, because there is a big difference between this and big difference between trial 62 or 63. So this is good and very interesting that now the awake replays are marking certain events. Now, the interesting thing comes, of course, is when we put the animal back into the home cage and the animal falls asleep, and we ask what information is being replayed during sleep ripples. And this is what we have done. And then it is the result, namely that what you can see, this is the awake replay that I showed you already. This is the distribution of the sharp wave ripples that you see, and each of these are replaying the previous experience. And the blue shows how replay during sleep weighs all these experiences. And you can appreciate that the two patterns are very, very similar, so much so that if you look at awake replay trial proportion with the post-sleep trial proportions, we got an astonishing, very high correlation. Now, in neuroscience, we are typically very happy when we got a 0.05 correlation. This is 0.86, extraordinarily powerful correlation. What it means is that during learning, that there is data activity, the differences from trial to trials are marked a little bit. The animal is rewarded. There are sharp wave ripples that selects the previous information and weighs it. If that is judged by the brain that is important, there will be a couple of or several sharp wave ripples. If it is judged that it's not so important, you won't have any. And the same memories or the same participants that have been participating in the awake sharp phase will be replayed hundreds and thousands of times during sleep for memory consolidation. So what I've showed you, that there is a selection mechanism. It's a very simple, mundane population burst called sharp wave ripple, and that gives us the opportunity as well as the favor that we don't have to absorb everything from the world, but certain things will be selected for us subconsciously because the subconscious brain is honest, is less biased. It contains all the previous experiences that we have, and it's probably much more effective than consciously thinking about many things. And those things that are just not so important, they go to the garbage can. So in summary, I remember this gentleman here, who I really, really, really admire, is because my brain generates sharp waves. And after the experience with Roy, many times that we have a conversation or a dinner, I had many opportunities to consolidate whatever I learned. If you are interested in more details, I have written a book about many of these things, and here is the Chinese translation. And I'd like to thank you.